Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 1 for October 2019. This is the Part 2 video. I'll put a link to the Part 1 video below in the description box. Let's begin with question 5. Question 5. An amoeba is a single-celled organism that lives in water. The photograph shows an amoeba as seen using a light microscope. So this is the amoeba. We have the nucleus, there is the cytoplasm, the cell membrane, as well as the pseudopodia. So they say, substances in the water can enter the amoeba by a variety of transport mechanisms. The table shows some features of transport mechanisms. They ask which feature is true for active transport only, facilitated diffusion only, both active transport and facilitated diffusion, or not true for both active transport and facilitated diffusion. So we have the feature as well as the transport mechanisms. For passive transport, we know it's by facilitated diffusion only, so this should be your answer. For membrane proteins involved, this should be for both active transport and facilitated diffusion, and that should be your answer. Direction of transport can be up the concentration gradient, that is for active transport only, and this should be the answer. Moving on. Here they say, the graph shows the uptake of two substances. There is substance K and substance L into the cytoplasm of an amoeba in water at a temperature of 18 degrees. On the vertical axis, concentration of substances in the cytoplasm. On the horizontal axis, we see time in hours. They want us to explain the differences in the uptake of substance K and substance L. So here we can see substance K is taken up really fast and it does not reach a constant level, while substance L is taken in at a slower rate and it reaches a constant level at around this point here. At this point, we can see equilibrium is established, meaning the concentration on either side is going to be the same. So my answer was substance K is taken up by active transport and substance L is taken up by diffusion. The reason as to why I chose diffusion is because equilibrium is reached. We can see the concentration becomes constant, and therefore that should be by diffusion. Down here they say, on the graph, draw a line to show the uptake of substance L at 10 degrees Celsius. If we go back to this experiment, we know it was carried out at 18 degrees. So carrying out the same experiment at a lower temperature means the rate of diffusion is going to be slower, because there will be lower kinetic energy and the particles are not going to have more successful collisions. So my line was beginning from the same concentration but at a lower rate, and the constant level was reached a little bit later on than this time here. Moving on. Here they say, pseudopodia are formed by cytoplasm flowing towards the membrane and changing its shape. Explain why the membrane is able to change its shape when cytoplasm flows towards it. Here I said the membrane is fluid, and of course you know the membrane is made out of phospholipids, it's made out of proteins as well as cholesterol. So fluidity is made by a balance between the cholesterol, the phospholipids, as well as the proteins. So here I say the membrane is fluid, the phospholipids and proteins make the membrane to move, and cholesterol helps to make the right fluidity. So next they say, explain how the uptake of substances would be affected if the amoeba increased its number of pseudopodia. If the number of pseudopodia increase, then there is going to be greater surface area, and therefore more absorption will take place, and a faster rate of diffusion will be carried out or will be observed. So here I said more pseudopodia increase the surface area, and therefore diffusion occurs faster. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Errors in DNA replication can give rise to mutations. The diagram shows the bases in a length of DNA. This is the length of DNA, and the corresponding bases are numbered. So this is number 1, and that is number 18. So they say the table shows the genetic code for the amino acids. Remember, the DNA is read in a codon, meaning 3 bases at a time. ATG is methionine, you can see there. I tried to name each of the 3 pairs of bases to give the corresponding amino acid. So down here they say the genetic codes TAA, TAG, and TGA are stop codons. State the sequence of the first 4 amino acids coded for by this length of DNA. As you can see, ATG is methionine. CTC, you come down here. This one is going to be leucine. And then isoleucine. Then we have that, and that. So I named them here, methionine, leucine, isoleucine, and tyrosine. So this should be the answer. Moving on. Here they say, a change in a single base can cause a change in the amino acid sequence produced in protein synthesis. Name the type of each mutation described below. When base number 3 becomes cytosine, means the base has been substituted, so this is going to be a substitution mutation. And when base number 6 becomes base number 5, it means one base has been removed, and therefore this is a deletion mutation. When base 9 becomes base 10, it means a base has been added, so this is insertion mutation. Down here they say, explain the possible effects of these 3 types of mutations on the amino acid sequence coded for by this length of DNA, meaning by this length, so we have to use examples based on this DNA sequence they've given us. And they say use the information in the table to support your answer. In my answer, I wrote about the different mutations and explaining their details. So I said, in substitution mutation, only a single base in the triplet codon is changed. This may lead to presence of a different amino acid in the protein. However, if the 6th base in the codon is the only one changed, then the amino acid may remain unchanged. I'm talking about the 6th base in the DNA codon they gave us. If the 6th base is the only one changed, then we're going to have CTA, CTG, and CTT, and all these will be coding for leucine. So this is an example in which a substitution mutation may not cause any significant difference in the sequence of amino acids produced. Now, in a deletion mutation, removal of a single base shifts all the bases by one position, and that means all the amino acids after the point of deletion are going to be affected. The example I used here is if you delete base 6, it leads to the combination called methionine, leucine, phenylalanine, threonine, and then isoleucine. So this is going to be a combination of different amino acids, and therefore the protein produced is going to be different. And lastly, in insertion mutation, the inserted base shifts the frame forward by one place. An example, if you insert G in the place of the base 6, this leads to methionine, leucine, histidine, leucine proline, as well as serine. So there will be different combinations of amino acids, and that will lead to a different protein being produced. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Blood flow and blood pressure are affected by a number of factors. Abnormal blood flow and abnormal blood pressure affect the health of a person. The graph shows the systolic pressure and diastolic pressure as blood flows through human blood vessels. On the vertical axis, we see pressure in kilopascals, and on the horizontal axis, we see the types of blood vessels. If I zoom out for you to see clearly, we can see the systolic pressure is quite high and the diastolic pressure is quite low in the different blood vessels, but when we go to the capillaries, they're going to be the same into the venules and into the veins. So let's continue to the questions. Here they say, pulse pressure is the difference between systolic pressure and diastolic pressure. In a healthy person, pulse pressure should be at least 25% of systolic pressure. Determine whether this person is healthy using the point indicated by the arrow on the x-axis. So if I take you back to the graph and looking at this specific point here where the arrow is positioned, we can see that the systolic pressure is about 15 kilopascals and the diastolic pressure is about 9.7 kilopascals. So I'm going to find the difference between them on the next page. So getting the difference, which is the pulse pressure, it's going to be 15 minus 9.7, which is 5.3. To get the percentage of pulse pressure, it is 5.3 divided by 15 times 100, which gives us 35.3%. And we can see 35.3% is greater than 25%. So we can conclude that the person is healthy because the pulse pressure is greater than 25% of the systolic pressure. So moving down here, they say the mean arterial pressure can be approximated using the formula MAP is equal to diastolic blood pressure plus systolic blood pressure minus diastolic blood pressure divided by 3. They say estimate the MAP in the elastic arteries for this person. We already have all these results, so I just substituted in here. Diastolic blood pressure is 9.7 plus systolic 15 minus 9.7 divided by 3, and my answer was 11.5 kilopascals. So that's the answer here. Moving on. Here they say, if the mean arterial pressure, MAP value, is less than 8 kilopascals, the force pushing the blood through the vessels will be too low. Explain how a low MAP could affect a person. If the mean arterial pressure is low, then it means blood is sent through the arteries at a lower pressure, and it means we cannot be able to deliver oxygen and nutrients in the right amount of time for aerobic respiration to occur, and therefore the cells are going to be low on ATP. So I said, at low pressure, blood will not reach the tissues in the organs at a fast enough rate, so the tissues will lack enough oxygen and nutrients for aerobic respiration. Less ATP will be available for cellular activity. When we move down here, they say, blood flow depends on the change in pressure, the radius of the blood vessel lumen, the number of blood vessels, and the viscosity of the blood. Then they say blood flow can be calculated using the formula. Blood flow is equal to that. And also blood flow can be calculated using this formula here. This involves resistance as well as change in pressure. So continuing on here, they say which formula can be used to calculate resistance. Now, since that and that are both equal to blood flow, I equated them in order to make resistance the subject. So resistance is going to equal to that times that divided by that, which we see here. And then similar terms are going to cancel out. That cancels out with that. And then we get that as the answer. So we look for something that has that combination. And here the answer is going to be a D. Then next they say, a change in which factor has the biggest effect on resistance? Now, if we look at the formula for resistance, we can see there is an R power 4. It means any change in this value is going to greatly affect whether there is an increase or a decrease. It's going to greatly affect the resistance because it's to the power of 4. So my answer was C because it has a power of 4. And then explain how arteries are adapted to reduce resistance to blood flow. To reduce resistance, they should be smooth or they have a smooth endothelial layer. So I said they have a smooth endothelium, and this reduces friction when the arteries are in contact with the blood. Moving on. Part C says, blood vessels can expand to accommodate increased blood flow. This is known as compliance. Explain how arteries are adapted to accommodate sudden increase in blood flow. If you remember, the arteries have smaller lumens. Arteries have a lot of collagen that allows them to withstand the changes in pressure. So I said, they have elastic fibers inside the arterial walls, and these fibers can be stretched so the diameter can be varied to widen the size of the lumen. These arterial walls contain collagen that has great strength to withstand the high pressure. And next here they say, explain why a reduction in the compliance of a blood vessel can lead to cardiovascular disease. If there is reduction in compliance, then it means there is going to be damage to the endothelial layer, and this could lead to the deposition of cholesterol and other ions, leading to formation of atheromas. So I said, a reduction in compliance can cause damage to the endothelial lining in the blood vessels. Cholesterol and ions can build up at the damaged site, leading to plaques or atheroma, and this blocks the blood vessels. The heart muscle may not get enough oxygen and nutrients for aerobic respiration, so muscular contraction may stop. So this brings us to the end of question 7. Let's continue to question 8. Question 8. The amino acid aspartate is converted into the amino acid arginine in the urea cycle. The urea cycle involves several enzymes and takes place in the liver. The diagram shows part of the urea cycle. So here we have ammonia, we have carbamyl phosphate, and so many components here and so many enzymes that participate in specific reactions. So on the next page they say, the R group of aspartate is as shown here. Draw the structure of the amino acid aspartate. Let me first delete this. Of course, if this is the R group, we just connect a carbon and then the carboxylic part this side, and then the NH2 this side and the hydrogen. So this gives me my structure you see here, and that should be the structure of the amino acid. Then next here they say, explain why several enzymes are involved in the urea cycle. Remember, enzyme-controlled reactions are specific in nature, so each enzyme and the substrate are complementary. The different substrates cannot bind into the active sites of just one enzyme, so each step is specific because they have different shapes. So I said, because the cycle has many stages or many reactions per stage, the product from each step is a substrate in the next stage, and each substrate requires a specific enzyme. Each substrate binds into the active sites of the specific enzymes in a lock and key model for a product to be formed. Moving on. Here they say ornithine transcarbamylase, or OTC, is one enzyme involved in the urea cycle. Ornithine transcarbamylase deficiency is an inherited genetic disorder. Suggest how a person can be shown to have this disorder. If a person has a genetic disorder, they could take a blood sample and check for a specific component. If we go back to the drawing, we can see that this is what we're talking about. There is deficiency of this. So they could measure the concentration of this from the blood and see if it's really high, then that means this person does not have the enzyme to break down this or to incorporate this into the ornithine cycle. So I said, with a blood test for a molecule like carbamyl phosphate, if in high concentration, it means none is broken down to urea. Also, they could carry out genetic screening to check for a mutation in the gene coding for OTC production. Or they could make a pedigree diagram to study the family history for presence of OTC deficiency. Moving on. Next they say, targeted messenger RNA therapy is being developed to treat OTC deficiency in mice. Targeted mRNA therapy involves injecting mice with phospholipid particles containing OTC messenger RNA. These particles, shown in the diagram, target liver cells. So we can see this is the messenger RNA. These are the phospholipids, and if you can see, it's like a bilayer, so it can incorporate well by fusing with the membrane of the cell. So here they say the table shows the results of one study using these particles. For mice, normal with treatment, the concentration of ammonia was 50 micromole per decimeter cubed, and we see this is for the normal concentration. With OTC deficiency and without treatment, we see the ammonia concentration is quite high. Then with OTC deficiency and with treatment, the concentration is 40 micromole per decimeter cubed. And as we observe, this is closer to the normal, which is 50 micromole per decimeter cubed. Moving on. Here they say, explain how this treatment works. They want you to use the information in the diagram of the urea cycle, the diagram of the particle, and use the table of results to support your answer. Now, using the table of results, we saw that mice treated with OTC messenger RNA had ammonia concentration of 40 micromole per decimeter cubed, and this was closer to the normal ammonia concentration of 50 micromole per decimeter cubed. Now, from the diagram, lack of OTC leads to the buildup of carbamyl phosphate because the carbamyl phosphate will not be incorporated into the ornithine cycle, so the concentration of ammonia increases. If I take you back, this is what we're talking about. If this is not there, that is not going to be incorporated into the ornithine cycle, and that means the ammonia concentration is going to be increased. Because even if we have this enzyme, that cannot be incorporated, so there is going to be a high concentration of ammonia because this is also in high concentration. So when we continue, then my next point was the phospholipid particles target the liver cells because the ornithine cycle takes place in the liver. That's true. So the phospholipids containing the messenger RNA will fuse with the liver cells. Remember, the phospholipids were also bilayers, so they can fuse and then deliver the messenger RNA into the cell. Then the phospholipids protect the messenger RNA from being broken down by hydrolytic enzymes. And lastly, the messenger RNA is translated into the functional OTC enzyme. This enzyme then incorporates carbamyl phosphate into the ornithine cycle by combining the carbamyl phosphate with ornithine, and this reduces the concentration of ammonia. This last point is about the mechanism of action and how, once this messenger RNA is delivered, how it helps to reduce the concentration of ammonia. So this brings us to the end of question 8, as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.